Dear students, now we are going to discuss photophosphorylation with the help of Peter Mitchell's chemiosmotic mechanism. After that, we will look into redox potential values, electron negativity. So before that one, we have to know how ATP is synthesized by means of non-cyclic electron transport system. To understand the mechanism of synthesis of ATP during non-cyclic electron transport system, we have to look into or study Peter Mitchell's chemiosmotic mechanism. With the help of that, we can understand what about the mechanism of synthesis of ATP. Chemiosmosis. By Mitchell. Peter Mitchell. So before this, we completed the mechanism of synthesis of NADPHH plus. And what about ATP synthesis? So this mechanism, chemiosmosis, explains, it is concerned with, it explains mechanism of ATP synthesis. ATP synthesis from ADP and inorganic phosphate. Okay, so that mechanism is called as a phosphorylation. Phosphorylation. Okay, so here, this mechanism, Peter Mitchell's chemiosmotic mechanism, explains a phosphorylation. What type of phosphorylation? In this case, here, it explains, first one, photophosphorylation. Photophosphorylation. So what is a photophosphorylation? Synthesis of ATP in the presence of sunlight. Synthesis of ATP from ADP and inorganic phosphate in the presence of light. Hence the name photophosphorylation. Okay, so in the case of chloroplast, are membranes which are able to perform photosynthesis, they are with a photophosphorylation. That photophosphorylation is of two types. One, non-cyclic photophosphorylation by means of Z scheme. And second one, cyclic photosphorylation. So these two are types of electron transport system during light reaction. Light reaction takes place with the help of two types of electron transport systems, non-cyclic and cyclic. So synthesis of ATP by means of non-cyclic is called as non-cyclic photophosphorylation. If it is by means of a cyclic one, cyclic photophosphorylation. We have to study this one. Leave it, don't worry. So overall, Peter Mitchell's chemiosmotic mechanism is concerned with the study of synthesis of ATP during light reaction in the presence of light. And the second one, oxidative phosphorylation. Oxidative phosphorylation. So oxidative phosphorylation means synthesis of ATP from ADP plus inorganic phosphate during oxidation of food. During oxidation of food. Okay, so oxidative phosphorylation during respiration. During respiration. So photophosphorylation within chloroplast, within thylakoids of chloroplast, within thylakoids, and in the case of cyanobacteria, chromatophores, go back to first year, chromatophores, in the case of cyanobacteria, and some bacteria also, but that was not given. Okay, so within the thylakoids of eukaryotes, chloroplast, chromatophores are present in cyanobacteria. Right? Next, oxidative phosphorylation during respiration. So this one takes place within mitochondria, particularly mitochondrial crestae of eukaryotes. Okay, or in the mesosomes, lateral mesosomes. First year part, linking mesosomes. Go back to characteristics of mesosomes. Mesosomes are supposed to involve in respiration. It was given there. Electron transport system. Okay. So simply, Peter Mitchell's chemiosmotic mechanism explains the mechanism of synthesis of ATP during light reaction, photophosphorylation and oxidative phosphorylation. Only these two. So this mechanism is not concerned with NADPHH plus synthesis. Only synthesis of ATP during photophosphorylation, that is a non-cyclic and cyclic, and during synthesis of ATP during respiration, oxidative phosphorylation. So definitely they will question you. Chemiosmosis is concerned with photophosphorylation and oxidative phosphorylation only. Synthesis of ATP during light reaction, that is photosynthesis, and synthesis of ATP during respiration, that is oxidative phosphorylation only. So this mechanism is not concerned with the synthesis of NADPHH plus. Remember that one. Okay. So now we are concerned with photophosphorylation only, not oxidative phosphorylation. We will study this one at the time of respiration, not now. So now take a photophosphorylation. So photophosphorylation takes place by means of ETS, electron transport system, that may be non-cyclic or cyclic. Respectively, they are called as non-cyclic photophosphorylation. Second one, cyclic photophosphorylation. Now, what about the mechanism of so-called chemiosmosis? So what about the mechanism? Before that one, requirements for chemiosmosis. So what are the main requirements of chemiosmosis? One, membrane required. membranes. And second one, H plus gradient. H plus gradient, proton concentration gradient. And third one, ATP synthase complex. ATP synthase complex. Membrane, whatever the membrane, we'll see what is that membrane. Proton concentration gradient is well known to you. Now, what is the ATP synthase complex? Okay, so what is a ATP synthase complex? Okay, so this is also called as CF0, CF1. What is a C and F? Coupling factor. CF, coupling factor, C, coupling F factor, CF0, CF1. So ATP synthase complex is also called as a CF0, CF1 complex. CF means a coupling factor, coupling, able to couple. We'll see what is that coupling. Remember this, the nomenclature of this one. So this ATP synthase complex, this complex is nothing but it is a protein, transmembrane protein. Transmembrane protein. What is that transmembrane protein? Simply, it is intrinsic protein. Intrinsic or integral. Go back to plasma membrane, first year. Integral protein. Okay, so according to fluid mosaic model, there are two types of proteins on the basis of ease of extraction. First year part. Okay, so intrinsic, extrinsic proteins, right? Intrinsic and extrinsic proteins. So this protein is a intrinsic protein and it is partly embedded, not completely embedded, partly embedded within the membrane, not completely inside the membrane. Some part is inside, remaining is exposed, protruded out. We will see what about that arrangement in chloroplast and mitochondria, right? So here CF0 is within membrane, not exposed. And it is a proton channel concerned with transportation of protons, proton channel proteins. Okay, F0 is within the membrane, integral part, embedded part. Okay, CF0 is a proton channel concerned with transportation of protons from one location to other location. That is also protein only, some part of protein, F0 inside the membrane. And this one is concerned with facilitated diffusion. We'll see what is that facilitated diffusion. Facilitated diffusion of H plus. Okay, and that is a non-catalytic, not concerned with catalysis. So this one is non-catalytic. I will explain again with the help of our diagram also. First description. So CF0 completed. Next one is a CF1. CF1. So CF0 is a stalk part actually. CF0 is stalk part, stalk like. And what about a CF1 head part? Head part. protruded into stroma in the case of chloroplast. In the case of mitochondria, protruded into mitochondrial matrix. We will study that one there, not here. So in chloroplast, head part is protruded into stroma. In mitochondria, it is protruded into mitochondrial matrix. So within CF1, one enzyme is present, that is ATP synthase enzyme. What is the role of this enzyme? It is able to synthesize a ATP molecule from inorganic phosphate and ADP with the help of energy. We will see what is that energy. So that's why CF1 is a catalytic part. So this part is a catalytic part. Catalysis, as it is having enzyme that is able to club. Actually, this one is called as coupling factor. What is the role of that one? Coupling of a inorganic phosphate and ADP. As these two are coupled, the complex is called as a coupling factor, CF0, CF1. Right? So what are the requirements? Membrane required, H plus gradient, ATP synthase complex. Now, what about the diagram for ATP synthase complex? For example, this is a membrane. Here I'm taking chloroplast, thylakoid membrane, both grana and stroma thylakoids. So this part, this one is CF0, completely inside the membrane, not exposed to stroma. For example, this is stroma lumen within the thylakoid membrane, both grana and stroma thylakoids. Okay, right? Next, the upper part, this one is head part. This one is head. So this one is a CF1. So CF0, CF1. So this is catalytic part, non-catalytic part. So now, what about the role of this one? For example, 2H plus passing through this one, passing through this one. So 2H plus. So what is the role of a CF0? Transportation of protons. I will explain mechanism again after completion of this one. So transportation of protons from lumen to stroma. So this one is concerned with a facilitated diffusion. More number of protons to less number of protons. Next, what about the role of CF1? Here enzyme is present. During transportation of protons, some amount of energy is liberated. That energy is used by ATP synthase enzyme to club ADP plus inorganic phosphate to ATP molecule. Okay, right? So that is about a ATP synthase complex. Right? Now, what about the mechanism? So these are the requirements. Now, what about a chemiosmosis mechanism? Now, according to your textbook, this is a transfer section of a granum thylakoid. Transfer section of a granum thylakoid. Okay, so here 680 is present. 680. right? So LHC of that one. Radiant energy absorbed by LSC. This is a ETS, non-cyclic electron transport system. So energy is reaching the 680. Now it is excited. Electrons are accepted by first electron acceptor, that is a pheophytin. So now electrons are donated to, given to PQ or not? Plastoquinone. PQ is not an electron carrier. It is a H plus transporter. So it was given like that. So for example, here I'm taking one electron accepted by that one. So here H2O molecule photolyzed. From the photolysis of a water molecule, half O2, 2H plus already available from that H2O molecule. Next, 2 electrons. So this is an electron transport system only, right? Now, PQE is transferring its electron to next electron acceptor. What is that one? Cytochrome B6 by F, cytochrome complex. So while transferring electron to that, reduced form of PQE is able to transport one proton into lumen. This is the lumen part. This is the lumen and stroma part. Stroma. And this one is a lumen, tube, transfer section of tube. So this is the lumen part, the empty place of that tube, right? One electron, one H plus, two electrons, two H plus. Take one more electron, two H plus, right? Now, plastocyanin, flow of electrons. Next, 700, ferridoxin. Next, finally, NADP reduced into NADP H plus, 2H plus from the stroma and 2 electrons from H2O, right? So here the question is, what is chemiosmosis? So chemiosmosis explains, in this case, the synthesis of ATP during non-cyclic photophosphorylation. Okay, so here we are within non-cyclic photophosphorylation. So now, less number of protons, more number of protons. So here within the stroma, less H plus. Less H plus means what about pH value? More. Negative logarithm of hydrogen ion concentration. What about pH here? Less. Reason? More number of protons. So less number of protons, more number of protons. More H plus. Go back to requirements of chemiosmosis. Membrane required. This is a thylakoid membrane. Next, proton concentration gradient. Less, more. So now, electrochemical potential gradient created. Okay, electrochemical potential gradient. Less H plus means more negative. More H plus means less negative or not. So more number of protons, less number of protons in the membrane, stroma, sorry. Okay, more number of protons. So now this membrane is impermeable to protons. But at certain areas, at particular areas, for example, here I'm taking that membrane is with a ATP synthase complex. ATP synthase complex. Okay, so this is ATP synthase complex. Just we discuss now. This one is a CF0, this one is a CF1. So this membrane is concerned with the transportation of a H plus from lumen to stroma, coming backflow of protons. Backflow of protons because of a proton concentration gradient. So now gradient is established, H plus gradient is established between stroma and lumen across the membrane. So more number of protons, less number of protons. This is a stroma part around the thylakoid. Stroma part or not? So this part is exposed to stroma. So now protons are transported from more to less. Here energy is not required for transportation of protons. So more to less is facilitated diffusion through the protein. This is a carrier protein. This carrier protein is able to transport protons from a region with more number of protons to the region with less number of protons without consuming ATP molecule. So this is a facilitated diffusion, right? So during transportation of 2H plus, for example, 2H plus, some amount of energy is liberated here. Come back. So this one is a ATP synthase complex, head part. So this one is a CF0, this one is a CF1. So now 2H plus transported from lumen to stroma. So during transportation of H plus, some amount of energy is liberated from the protons. So some amount of energy liberated from the transportation of so-called H plus, some amount of energy. That energy is coming from sunlight. So this energy is used by ATP synthase enzyme to club ADP plus inorganic phosphate into one molecule of ATP. So during transportation of protons from a region with more number of protons to a region with less number of protons means lumen to stroma. So lumen is acting as a reservoir of protons. It is a continuous process. So continuously more number of protons from here to here, continuous process. So while transporting protons, that CF1 as it is catalytic part that is able to utilize energy coming from transportation of protons to synthesize a ATP molecule from ADP and inorganic phosphate. So according to Peter Mitchell, for transportation of 2H plus, number of ATP molecules synthesized is only one ATP molecule. So 2 is to 1 ATP molecule. Okay, so go back to chemiosmosis. According to chemiosmosis requirements, membrane required, proton concentration gradient, ATP synthase complex. So this complex is able to break a proton concentration gradient by transporting protons from lumen to stroma. So if proton concentration gradient is broken, ATP synthase is able to synthesize ATP molecule. So what is the role of this protein? Transportation of protons, breaking the proton concentration gradient, and the synthesis of ATP molecule out of the energy coming from transportation of H plus. So here, simply I will give you, wait. So this is given in your textbook. I will give you simplest one. Very simplest one. For example, this is a thylakoid membrane, granum thylakoid. Yesterday we took granum thylakoid one. Take as is that one, granum thylakoid here. So this is a Z scheme, non-cyclic photophosphorylation. So take now one H2O molecule. One H2O molecule is photolized. 2 electrons, 2H plus. So photolysis of water is within the lumen. This is lumen part, lumen. And this one is thylakoid membrane. And this one is a stroma. Stroma, that is matrix of the chloroplast. 2 electrons, 2 H plus, and half O2. So half O2 diffuses from lumen to out. Lumen to out. That will be released into atmosphere. So now, what about the number of H plus available? 2 H plus. So now, these 2 electrons transported through various electron carriers like that. Okay, 680, pheophytin. I don't want to use those terms here. Already yesterday completed. 680, pheophytin, PQ, cytochrome complex, plastocyanin, 700, pyridoxine. So finally, NADP plus is reduced into NADPH H plus. So how many electrons? 2 electrons and 2 H plus from the stroma with the help of some enzyme. What is that enzyme? FNR, pyridoxine NAD reductase or NADP oxidoreductase. So NADPH plus synthesized. Now, what about chemiosmosis? See here, how many H plus? 2 H plus. During transportation of electrons, how many electrons? For example, 2 electrons. What about the number of protons translocated? 2H plus. So yesterday I asked one question. What about the total number of H plus within the lumen? So how many? 4 H plus. Here less, more. It is a continuous process. Less number of protons, more number of protons. Okay, so now take ATP synthase complex here. This one is a ATP synthase complex. F0, F1. Okay, so CF0, CF1, coupling factor. So according to now, Peter Mitchell's chemiosmotic mechanism, breaking of proton concentration gradient. What is that breaking? From lumen to backflow of protons from lumen to stroma, 2H plus. Okay, so proton motive force, proton mobilizing force, proton mobilization force or not? Okay, so for example, negative charge more here, negative charge very less here, or positive charge more here. So definitely to balance charge, protons must be transferred from lumen to stroma. That is according to electrochemical potential gradient. So out of 4H plus, take 2H plus out of 4, one by one. To understand the concept, I'm giving like that. 2H plus transferred, again 2H plus to understand the concept only. So according to the concept, what about the number of ATP molecules synthesized? 1 ATP molecule from ADP plus inorganic phosphate. So like that, how many H plus available? 4. So 4 divided by 4H plus divided by 2H plus. How many ATP? 2 ATP molecules. Okay, so ratio is a 2H plus 1 ATP molecule. According to that ratio, out of this much number of H plus transported through ATP synthase complex, definitely how many ATP possible? 2 ATP possible. Okay, so 4H plus available. How many ATP? 2 ATP molecules. So now, out of one H2O molecule photolized, what about the number of ATP molecules? 2 ATP molecules. How many NADPHs plus? 1 molecule. So during, this is very simplest one. Okay, so what about the place of synthesis of NADPH within the stroma? Synthesis of ATP within the stroma. So now NADPH plus and ATP are available within the stroma to run Dark reaction. Definitely they will question you. What about the place of formation of NADPHS plus and ATP? Go for stroma only, don't go for membrane, right? So out of one H2O molecule, how many ATP produced? 2 ATP. How many NADPHS plus? 1. So according to that statistics, now what about a total number of ATP and NADPHS plus possible out of 12 molecules of ATP photolysed? If you go back to oxygenic photosynthesis, 6CO2 plus, here given one is 12H2O. This is very, very important. One question was given, what about the use of a 12H2O molecule in that equation? What is that role? We'll see. Glucose molecule plus 6H2O plus 6O2 molecules are 12 half O2 atoms. Now take a 12H2O molecules. Okay, so non-cyclic electron transport system. Here I'm taking only H2O molecules. I don't want to take any electron carriers. You use them, please. So take now 12 H2O molecules. 12 H2O molecules photolysed with the help of OEC complex. Okay, so what about oxygen and electrons? So 12 H2O means 12 half O2. So 12 half O2s are 6 O2 molecules. 12 half O2 atoms are 6 O2 molecules. So how many H plus? 12 into 2. 24 H plus directly coming from photolysis of water. How many electrons? 12 into 2. 24 electrons. 24 electrons. Now, these 24 electrons passing through 680, pheophytin, PQ, and continue it. Okay, so how many electrons available? 24 electrons. 24 electrons plus 12 molecules of NADP plus, that is in oxidized condition, able to accept electron. That is very, very important. After this, we have to know what is electron negativity or redox potential values. So 24 electrons, sorry. And from stroma, what about the number of H plus? 24 H plus coming from stroma. With the help of a concerned enzyme, that is NADP reductase. Product is 12 molecules of NADP H plus. Okay, so ratio between H2O molecules oxidized, number of NADP H plus produced is 1:1 ratio. Okay, now during transportation of 24 electrons, what about the number of protons translocated or transferred or removed from stroma and added to lumen part? 24 H plus. Now all together, how many? 48 H plus. 48 available out of a 12 H2O molecules photolysed. Now F0, F1 complex. CF0, CF1 complex. So how many protons available? 48. 48 H plus. So according to that calculation, 48 divided by 2H plus. How many ATP? 24 ATP molecules. Okay, so in this case, to produce a 24 ATP molecules, 24 ADP, 24 inorganic phosphates are required. So if 12 H2O molecules are photolysed during non-cyclic electron transport system, what about the energetics of this non-cyclic electron transport system? So 12 H2O molecules. 12 H2O molecules. How many ATP molecules? 24 ATP molecules. How many NADPH plus? 12 molecules of NADPH plus. I will write separately, right? So non-cyclic electron transport system or photophosphorylation accounts for synthesis of ATP and NADPH plus also. These two are collectively called as reducing power. So during non-cyclic electron transport system, out of 12 H2O molecules photolized, so here I'm separately giving Z scheme. In Z scheme, if 12 H2O molecules are photolized, oxidized, what about the number of NADPH plus? 12 molecules of NADPH plus. And how many ATP molecules? 24 ATP molecules. And what about a number of oxygen molecules? 6 O2 I'm taking, or 12 half O2s. Right? From that, how many glucose molecules are produced? One glucose molecule. Okay, so one H2O molecule. H2O molecule ratio ATP and NADPH plus. Here I'm taking one H2O, two ATP molecules, one NADPH plus. Okay, so non-cyclic electron transport system or Z scheme photophosphorylation non-cyclic accounts for synthesis of NADPH and ATP also. These two are collectively called as assimilatory power. So non-cyclic electron transport system is able to convert radiant energy into chemical energy. So during conversion of radiant energy into chemical energy, out of 12 H2O molecules photolized, for photolysis of water, so 12 H2O molecules needs how many photons? How much amount of energy is required? So it needs 12 into 4, 48 photons, not protons, energy particle, PHOTONS, photons. 12 H2O in the presence of this much number of photons, H2O, 12 molecules, is oxidized into 24 electrons plus 24 H plus and 12 half O2s atoms are 6 O2 molecules, right? So that is about a non-cyclic photophosphorylation, right?